Hello, my name is Professor Michael Walsh and I'm a biomedical engineer. In this project, I'm interested in mechanically characterizing the soft biological tissue that we're going to be working with. We're going to characterize that tissue from both a solid mechanics perspective in terms of how the tissue behaves, and we're also going to characterize it from a fluid mechanics perspective in terms of how the fluid forces affect the cells that are on the surface of the tissue. Once we characterize the tissue, we're going to decellularize the tissue using a custom-made rig that we have designed here. And following that, we will recharacterize this tissue scaffold to determine regional property variations within the tissue. Once we have determined these properties, we will pass this information on to the other members of the team who will use this information to impose these forces onto the cells.
Hello, my name is John Mulvihill and I'm also a biomedical engineer. My interest lies between the macro and micro level. For instance, looking at how physical forces affect cell function and behaviour. For this shared project, what I'll do is I'll take the physical forces that Professor Walsh has identified that occur in the intestine, and I'll develop a system where we can seed cells in a hydrogel, which is developed by Professor Eige, and mimic the forces that are in the intestine onto these cells. We can create a multi-layered hydrogel that looks at the different mechanical properties of the different areas in the intestine and seed the corresponding cells. We can look at how these forces then change and alter these cell types, going from quiescent to a reactive state.
Hello, my name is Jacques Heuger. I have more than 15 years of experience with the study of hydrogel and the application of hydrogel in the human body. In this project, I will apply my knowledge of hydrogels to the intestine, and I will use the data obtained from other colleagues, namely Michael Walsh and Kieran Magurti, on physicochemical and mechanical properties of the intestine to mimic the properties of the intestine using different hydrogels. I will develop a layered set of hydrogels which individually mimic specific properties needed for different cells into the intestine and apply these to grow cells onto these hydrogels.
Hello, my name's Kieran and I'm a biologist. So I'm interested in how stem cells respond to their environment. In this project in particular, I'm interested in identifying the biomolecules that govern this stem cell behavior in the intestine. We're going to use shotgun approaches like proteomics to identify these biomolecules. Once we've identified these biomolecules, I'm going to incorporate them into the material that Jack, John and Mika have created. We will then populate this scaffold with stem cells. And these biomolecules and the biomechanical properties of the material will induce these stem cells to differentiate into all the different cell types of the intestine. In this way, we can create an intestine outside of the body on an organ on a chip.
Hello, my name is Andreas Grapocker. I'm also a biologist and I'm interested in how the brain functions. In this project, I will explore how nutrients such as trace metals influence brain development. For this, they have to be absorbed across the gastrointestinal barrier before traversing the blood-brain barrier. Using our in vitro organ-on-a-chip mimics, we can model this process of absorption, reducing the need to do so in animals. Recently, we have shown that this process of trace metal absorption is critical for the development of brain disorders such as autism.
My name is Pat Kiley. I'm a cancer biologist working at Graduate Intro Medical School and here at the Bernal Institute. I'm really interested in how colon cancer and breast cancer develop and trying to come up with better ways to treat the disease. My work is focused particularly on figuring out how cancer spread. If we can come up with better approaches to figure out how cancer spread, that's going to give us the best way to fight the disease. And that's why I'm really interested in working with my colleagues here in the Bernal Institute to try and bring a multidisciplinary approach to what we're doing. If we can characterize the tissue within the tissue microenvironment, then we can come up with better ways to treat the disease and predict if these cancers are going to spread.